Thank you for tuning in to this health announcement produced by The Who. In today's video, we will be discussing mitosis and meiosis in its entirety, giving in-depth explanations on these two vital processes that regularly occur in the body. Dr. Crappen and myself, Dr. Culberson, will be your hosts today.
Let's start off with the basics of mitosis. Mitosis in itself is broken down into 6 stages. Its main purpose is to duplicate its DNA and create 2 identical daughter cells from the original cell. Its first stage is interphase. This is where the period of the cell cycle during which the nucleus is not undergoing division. One on the outside of the nucleus, 2 centrosomes containing centrioles, which are key for the process of the cell division. Next is prophase, the first stage in PMAT. In this stage, the chromosomes combine into an X-shaped structure. Each of these chromosomes are comprised of 2 sister chromatids. While that occurs, the membrane dissolves, releasing the chromosomes and letting the mitotic spindle to help stretch across the cell between the centrioles. Next is metaphase, the M in PMAT. In metaphase, the chromosomes line up at the center of equator of the cell, while the mitotic spindles sit on opposite sides, with the centrosomes basically pulling the chromosomes apart. After that, we have anaphase, the A in PMAT, where the chromosomes are torn apart by the mitotic spindle. Then, in the final letter in PMAT, the T for telophase, where a membrane surrounds around each separate chromosomes to create 2 nuclei. In the final stage, we have cytokinesis, where basically the single cell splits the 2 nuclei.
Next we have meiosis. In itself, it is broken down into 10 stages. Its main purpose is to lead the start of sexual reproduction, with one cell to be reduced to normal diploid cells, then to haploid cells called gametes, to help in the process of creating an organism. Its first stage is interphase. This is where the period of the cell cycle during which the nucleus is not undergoing division, while on the outside of the nucleus, 2 centrosomes containing centrioles, which are key for the process of cell division. Next is prophase 1, the first stage in PMAT. In this stage, the chromosomes combine into an X-shaped structure. Each of these chromosomes are comprised of 2 sister chromatids. While that occurs, the membrane dissolves, releasing the chromosomes and letting the mitotic spindle help stretch across the cell between the centrioles. The pairs of chromosomes are also exchange bits of DNA called recombination. Next is metaphase 1, the M in PMAT. In metaphase 1, the pair of chromosomes line up at the center of the equator of the cell, while the mitotic spindles sit on opposite sides, with the centrosomes all attached to 1 centrosome of each pair. After that, we have anaphase 1, the A in PMAT, where the chromosomes are torn apart by the mitotic spindle and the sister chromatids stay together. Then in the final letter in PMAT, the T for telophase 1, where a membrane surrounds around each separated chromosome located on the opposite poles of the cell to create 2 nuclei. And in the final stage, we have cytokinesis, where basically the single cell splits into 2 nuclei. Then we carry on to mitosis 2, where the second stages of PMAT occur. First is prophase 2, now with the 2 daughter cells containing 23 chromosomes again form an X shape. Then the membrane displaces and the centrioles duplicate and the mitotic spindle forms again. Then in metaphase 2, the 2 daughter cells line up again to the end along the center of the cell, and the mitotic fiber at each pole attached to each of the sister chromatids. Next we have anaphase 2 in PMAT 2. In this stage, the sister chromatids are torn apart by the mitotic spindle. Finally, telophase 2, where the chromosomes complete their move at each pole to gather a full set, forming into 2 new nuclei. To all sum up the elongated process is cytokinesis, to complete 4 granddaughter cells, each with a half set of chromosomes.
After those riveting prose explanations, there's still more to learn about mitosis and meiosis. First, let's get personal with mitosis. In the process, two haploid cells are created to be identical. In mitosis, there's always the same amount of chromosomes. Lastly, mitosis can create blood cells, muscle cells, but never get gamete cells.
Now to get down with meiosis. In this specific process, 4 gamete haploid cells are created to be different daughter cells. Critical things to know is that its cells are reduced by half, and begin with diploid cells that divide twice.
Now, because we got some basic knowledge about meiosis and mitosis, let's see what the differences are. Now, to start off with mitosis. So basically, mitosis is in somatic cell divisions, whereas meiosis is happening in germs. Furthermore, mitosis is used for growth and repair, and meiosis for making egg and sperm cells. However, one of the most significant differences is that mitosis goes through one stage and mitosis goes through 2 stages. Now you're probably all confused and ask yourself, why is that so important? Well, the process of mitosis is due to the one stage only having one copy of the DNA and one nuclear division leading to 2 identical daughter cells. On the other hand, meiosis has unbelievable 1 copy of the DNA and 2 cell divisions leading to 4 daughter cells. Moreover, there are still a few more differences, like meiosis has a sexual reproduction, whereas mitosis is asexual. Meiosis has different genetics and mixes the chromosomes, while mitosis has identical genetics and the stage of crossing over does not occur. When it comes to the aspect pairing homologs, meiosis does and mitosis does not.
But there are not only differences. Meiosis and mitosis also share many similarities. Both of these processes include diploid parent cells. They both consist of interphase, PMAT, and cytokinesis, which are stages of the process. In metaphase and metaphase 2, both these stages' individual chromosomes line up along the equator. In anaphase and anaphase 2, both these stages have sister cells that are separated to opposite poles.
But the biggest question is, what are the advantages and disadvantages of both of them? Well, mitosis is doing cell division and asexual reproduction, and the advantages from that is that it has quick reproduction. It needs only one mate, and last one at least, it only needs one parent. But there's also some disadvantages on there. Mitosis cannot adapt to change. There's no genetic diversity, and the DNA is exactly the same one as the one from your parents. After we clarified mitosis, now we can move on to meiosis. His advantages are that there's genetic diversity. It can adapt changes and the genes are changing each generation. However, even meiosis has disadvantages, which are slow reproduction, and it needs 2 parents, so a male and a female, that the process can be working. Thank you for watching our health announcement, and we hope that you have gained some knowledge on the concept of mitosis and meiosis. This has been Dr. Colberson and Dr. Conrad signing out.